Okay, we're going to go ahead and get started. So if I can have your attention, we're going to go ahead and get started. And we're going to continue on with our lectures about cancer and DNA repair. We're just going to pick up right where we left off on Friday. So just as a short reminder, just to catch up where we were starting to talk about some complex repair mechanisms that are involved with repairing DNA damage. And we started looking at mismatch repair. This is obviously designed to repair mispairs that are incorporated into the DNA damage. And that involves this protein called mut-S, if you remember. It looks somewhat like two hands reaching down, grabbing the DNA where the mismatch occurs, allowing other factors to cut the DNA and remove the mispairs and then replace it through DNA replication so that the mispairs can, or the DNA polymerase can get another crack at putting the correct nucleotides into the DNA. So now we're going to move on to another type of DNA repair called base excision repair. It's often abbreviated BER because it's a long name. But this, as the name implies, this type of DNA damage is involved in, actually I'm going to put this up into a slideshow format and get the pointer showing. So as I was saying, as the name base excision repair implies, this form of DNA repair involves cutting out specifically one base. And it's used to target many common forms of DNA damage, such as an adeoxoguanine, which we looked at earlier as a form of damage that results from free radical oxygens. So adeoxo-Gs are repaired by base excision repair. And the way this process works is that there are enzymes called glycosylases, which are designed to specifically recognize a certain type of DNA damage. So adeoxoguanine is recognized by an adeoxo-G DNA glycosylase. And it comes in and binds to this damage and cuts the base off from the nucleotide. So you're left with an abasic site. The sugar and the phosphate are still in the backbone of the DNA, but there's no base present. And then an endonuclease can come in and cut out that abasic site altogether. There's a free 3' OH on the left on the template or on the DNA strand. So DNA polymerase can come in, incorporate a nucleotide, the correct nucleotide, and then ligase can join those strands of DNA to complete a full double-stranded DNA without the damage present in the DNA. So as I mentioned, adeoxo-G DNA glycosylase specifically recognizes these adeoxoguanine damage. In bacteria, this is also called mut-M. So mut-S is involved with mismatch repair. Mut-M is involved with base excision repair. And it binds to these adeoxoguanines. And basically what it does is it takes the guanine that is damaged and flips it out of the double helix. So the way it works is if you have your base pairs, like this is your damaged guanine, and this would be the cytosine that's base paired to it. When adeoxo-G DNA glycosylase comes in, it basically flips the adeoxo-G out away from the double helix. And it does this by sticking an amino acid in place of the damaged guanine. So when that adeoxo-G is flipped out, there's certain residues that are able to hydrogen bond with this guanine, keeping it held away from the double helix. So you can see that there's a phenylalanine and a cysteine, which are very important, as well as this glutamine. And these all help to hold this adeoxoguanine so that the glycosidic bond between the sugar and the adeoxo-G can be cut and that base is removed. Now on the other strand of the DNA where the cytosine was base paired with the hydrogen, I said there was an amino acid that comes in and takes the place of the guanine. And that amino acid is an arginine, shown here as arginine 204. And it hydrogen bonds specifically with the cytosine so that it's not left unpaired and flopping around, available for other potential damage. And it stays there, holding that cytosine until the glycosylase accomplishes its work, and then replication can be done again to base pair with the cytosine. Another type of glycosylase is called uracil DNA glycosylase. This is designed to recognize uracils in DNA because uracils only belong in RNA. And if you remember, spontaneous form of damage is deamination of cytosine, which results in the formation of uracil. Uracils are also occasionally incorporated by DNA polymerase. Either way, they can be present in the DNA, and thus you need uracil DNA glycosylase to remove them from the DNA. And being an enzymologist, I have to include a little bit about the enzymes and how they actually accomplish their work. If you remember from your enzyme mechanism chapter, enzymes are involved with reducing the energy of the transition state to help the reaction to actually proceed. Well, uracil DNA glycosylase has a unique type of activity called substrate stabilization. And basically what this means is that the substrate itself, which is the DNA, provides that the activity or the energy to stabilize the transition state. And that's because in the structure, in the picture here, you can see a picture of this is the phosphate backbone of the DNA. Here's the uracil that was attached to this sugar. And uracil glycosylase serves to remove the uracil from the sugar. And as the sugar is leaving as the base is leaving, the sugar starts to develop a positive charge. Well, there's all these phosphates in close proximity to this positive developing charge. So it actually helps to stabilize it as as this sugar becomes more positive. And that's how the DNA serves to stabilize the transition state. And that's what substrate stabilization is. And that's utilized by uracil DNA glycosylase. Are there any questions about this? It's kind of a vague idea that we've hit on, but okay, I'm going to move on. And if I don't see a hand when it's raised, just yell out the question because there's a lot of people in here. So there are times that I might not notice a hand being raised. So just to review again, basic excision repair. The glycosylase recognizes a specific form of DNA damage and binds to it, removes that base, leaving the sugar and the phosphate backbone. And interestingly, the glycosylase stays there after the base is cut until the other endonuclease and other enzymes involved in the process can come and take over. Perhaps to serve as a signal to the other enzymes to come to the site of damage, or also just to keep that A-basic site somewhat protected from other side reactions that might occur. But either way, eventually AP endonuclease comes in and cuts out that DNA. AP stands for apurinic, and that's another name that is often used referring to an A-basic site. Now, bases can be purines or pyrimidines, but historically, A-basic sites are referred to as apurinic. So we have AP endonuclease. It cuts the DNA on the 5' side of the sugar, and it cuts out the sugar and the phosphate backbone, leaving this site to be replicated again by DNA polymerase and sealed with DNA ligase. Now, DNA polymerase may actually continue with replication for a short distance, producing a flap, again, an extra flap of DNA in this area, perhaps. So the ligase that might come in, or actually the endonuclease, flap endonuclease may be required to cut out that extra flap of DNA so that there's not this weird structure of a double helix with an extra little bit of DNA hanging out. So that's basically how base excision repair occurs. Now to move on to another form of DNA repair is nucleotide excision repair. And again, as the name implies, this process involves the removal of the entire nucleotide. Not only does it remove the damaged nucleotide, but it actually removes a pretty large segment of DNA on both sides of it. And nucleotide excision repair is very important for repairing UV damage, such as thymine dimers. If you remember, we looked at those as well. They result from UV light, where two adjacent thymines are cross-linked to each other and not easily handled by DNA polymerases. What happens in nucleotide excision repair is that the enzymes involved with recognizing the DNA damage will bind, and they recruit a helicase to come in and unzip the DNA around the damage. And then an endonuclease can come in and cut, as I said, a large fragment on each side of the damaged DNA. Usually it's about 30 nucleotides that it cuts out. So it's not huge, but it's pretty large, very large enough segment to cut out and be replaced. And then that's, again, filled in with DNA polymerase and DNA ligase so that the damage is removed and fully replaced with non-damaged DNA. Okay. Now, nucleotide excision repair, a lot of the enzymes involved with this process were first discovered through through studies of different diseases, actually. For example, Cockayne syndrome. Because of people who were studying Cockayne syndrome, we also were able to discover a lot about nucleotide excision repair. What Cockayne syndrome is actually a neurological development defect where individuals do not properly develop, and some signs of Cockayne syndrome include like short stature, they're really not very tall, they might have UV sunlight sensitivity, premature aging, their eyes might be sunken in, and actually pretty easily identified as someone who has Cockayne syndrome. And it was found that there are 5 genes that are responsible for this disease. So it's a genetic disease, and if there's a mutation in one of those 5, individuals will likely develop Cockayne syndrome. What these genes encode are proteins that are involved with recognizing RNA polymerases that are stalled at DNA damage. So I believe I mentioned that DNA damage isn't well handled by DNA polymerase, and it's also not handled well by RNA polymerase. And RNA polymerase during the course of transcription will also stall at the site of DNA damage. So as RNA polymerase is moving along at the DNA and encounters DNA damage, it will basically stall, just like a DNA polymerase would. While it's stalled, there's these other genes, these 5 genes that I mentioned, that recognize the stalled RNA polymerase, and they can come in and help to remove the stalled RNA polymerase and also recruit these nucleotide excision repair genes so that the damage can be replaced. So if these genes don't work, RNA polymerase is pretty much stuck there, unable to go on, and nucleotide excision repair cannot be accomplished either. So as a result, there's unrepaired DNA, and if it accumulates enough, it leads to apoptosis of the cells. So neurons might not be able to develop fully because of unrepaired DNA, and as a result, the neurons are actually apoptose instead of developing as they should. Another genetic disease that helped in understanding more about nucleotide excision repair is the disease xeroderma pigmentosum. This is abbreviated XP. And this disease is characterized by a highly increased sensitivity to sunlight. Exposure to UV should be minimized for anyone with XP because UV light will induce skin cancer on these individuals. Individuals with XP are 1,000 times more likely to develop skin cancer than anyone else around them. So you might have heard about individuals with XP before that basically have to live inside most of their lives because they don't want to be exposed to sunlight because of the skin cancer. Well, it turns out that there are genes, 7 genes directly involved with nucleotide excision repair, that if any of these are defective, they lead to the development of XP. That's because UV damage, which is caused by sunlight, such as thymine dimers, they can't be repaired. And so, again, other mechanisms, if the cell's left up to finding other mechanisms to deal with the UV damage, it often leads to development of cancer. So these 7 genes are responsible for nucleotide excision repair, but if they're dysfunctional, they're responsible for XP as well. Now, the kinds of XP that are known are abbreviated XPA through G. And it turned out that there was one more type of XP that was really quite puzzling to scientists for quite a while because it displayed all the normal characteristics of xeroderma pigmentosum, but none of the nucleotide excision repair proteins were altered. And it took quite some time before it was actually found out that this variant form, abbreviated XPV, is actually caused by mutation in a DNA polymerase. Actually, a scientist in my lab was involved with one of the two labs that simultaneously discovered that poly-A is involved with development of XP. And poly-A is the abbreviation for this DNA polymerase that is responsible for the development of XPV. So DNA polymerase eta is what's called a specialized DNA polymerase. It is specialized in its activity and how it replicates and what it replicates through. So it appears that eta is actually specifically designed to replicate through certain types of damage, such as 8-oxoguanines, which we looked at, as well as thymine dimers. And it replicates through both of these correctly. Essentially, it goes through these, we know from kinetic studies that poly-A can replicate through this damage without even really sensing that the damage is there. So that's its highly specific function. And if you remember about DNA polymerases, they have this tight active site where only the correct base pairing usually fits into the active site. Well, this is the structure of DNA polymerase eta, and you probably can't tell by looking at it, but the active site of poly-A is actually a lot more open and flexible and leaves room for potential distortions in the DNA and potential damage to actually fit into the active site, so it can still replicate through it. So we know structurally that's how poly-A actually does what it does. It's really quite amazing in its ability to replicate through this damage. So since it can replicate through thymine dimers, if it's not functional, if there's defects in the activity of DNA polymerase eta, it leaves a cell unable to replicate correctly through UV damage, such as the TT dimers. And again, it leads to the development of skin cancer. But while we're talking about specialized DNA polymerases, another term that's often used for them is translesion DNA polymerases. That's because a lot of these specialized polymerases function in replicating across DNA lesions. So trans means across, lesion obviously indicates damage in the DNA. So even though base excision repair and nucleotide excision repair work pretty well to remove a lot of damage from the cell, it's still inevitable that there's going to be damage in the DNA when replication is occurring. In fact, there's steady state levels of 8-oxoguanine. It's estimated that there's nearly 10,000 8-oxoguanines at steady state when base excision repair, in one cell, there's 10,000 8-oxoguanines, even when base excision repair is fully functional. So these damaged bases are going to be there, whether we want them to be there or not, during the course of replication. So that's why it's really good to have these translesion DNA polymerases. And it turns out that each one of these specialized polymerases has a specific function and replicates through specific types of DNA damage. Here I show the structure of a different specialized DNA polymerase called Rev1. This incorporates opposite A basic sites. When there's no base there, it'll incorporate a C every time. It also incorporates off of 8-oxoguanines as well, and some other more odd forms of damage that we haven't talked about today. And there's another one that actually utilizes Hoogsteen base pairing to deal with adducts that are on guanines, and so it uses non-canonical base pairing. It's just quite amazing at how well these have adapted to handle bulky lesions in the DNA. One side product, though, is that these translesion DNA polymerases can also be error-prone. So they might be able to replicate correctly through damage, but if they were to replicate the entire genome, they would actually end up making a lot of mistakes. So they need to have limited access to the DNA in order to accomplish what they're doing. Although, if you think about this from the perspective of evolution, errors in DNA can be good at times, because if evolution is to occur, some changes do have to occur in the genes at some point in order to allow a cell to adapt to a new environment. So errors can be good, and in that way, these polymerases might help to promote evolution. Also, it can also enhance survival rate during times of stress, say lack of nutrients or other other situations when the cell is stressed that help the cell to actually survive. It's better to survive than to die altogether. So again, error-prone polymerases can be very, very useful in the cell. Are there any questions about this before I move on? Okay. I'm going to kind of switch back to another form of DNA damage here. And these are gaps in the DNA. So what I mean by gaps in DNA are, say, a single-stranded break or a double-stranded break. You have a nick, or you can have complete breakage of the entire double helix. And these gaps are nearly unavoidable. Irradiation causes a lot of gaps in DNA, a lot of breakage of chromosomes in DNA. And other processes as well lead to the formation of gaps. So it's very hard to avoid them. DNA polymerase cannot replicate through gaps in the DNA, especially if the bases are missing altogether. There's no template to pair off of, so it can't go anywhere. And so it inhibits the process of DNA replication. And there's a process that is designed to facilitate replication even when there are gaps present in the DNA. So that's called recombination. And recombination can be accomplished through single-stranded DNA breaks or double-stranded DNA breaks. And I also mentioned here that end joining is another process that fixes double-stranded DNA. So we're going to look at both of these single-strand breaks and double-strand breaks. Both of these processes with recombination require that there be homologous double-stranded DNA template present in the cell as well. And the process of recombination is actually highly conserved between cells, all the way from bacteria to humans. They're all very, very similar in similar process. And it indicates that maintaining the integrity of the DNA is very important in all cells. And that's why we have recombination process still usable in human cells. So recombination with a single-stranded break is what we're going to look at first. So you might want to actually go back to your book and study these pictures quite a bit, because it takes a while to really understand what's going on if you look at the pictures. So I stared at the pictures for quite some time before I was able to understand exactly what's going on. But basically what's happening is here's your DNA that's going to be replicated. And here is, with the arrows, indicate the newly synthesized DNA. Turns out there's a base that's missing, so there's a gap in this strand of DNA. So as replication is proceeding, and this fork is opening up more and more, eventually the DNA polymerase on this strand is going to encounter the gap. And it can't go anywhere because there's no template. So what happens is that replication stops on that strand. It might continue for a little while on this strand, replicating through the DNA. But eventually a helicase will unwind the newly made DNA, leaving this strand of DNA actually unattached. And there's a free 3' end from this strand here that can basically invade the double-stranded region down here. So it comes in, and it can base pair with the other newly made strand of DNA. And DNA synthesis can occur for a short distance with that newly synthesized strand from the other side, or from the other strand of DNA. And thus it's able to utilize that DNA substrate for base pairing, so it can actually incorporate the correct nucleotide. And then after this replicates for a short distance, this whole complex structure of crossed over strands is going to be resolved, and you end up with base pairing back with the original template, and replication can continue as before, and you're still left with an abasic site that needs to be repaired in that DNA template. So that is recombination that bypasses a single strand gap of DNA. Recombination that's repairing a double strand of DNA, double strand breaks of DNA, requires another homologous DNA strand, as I mentioned earlier. Basically, if both strands of this, if this DNA is just cut full through, you have two strands of DNA. Both of them, the 5' ends, which would be represented here and here, need to be cut back so that there's a strand of 3' DNA that's single-stranded on both of those. And then utilizing the homologous DNA that's nearby, these 3' strands can invade and base pair with the appropriate strands for templates. Then replication can continue for a short distance, utilizing that homologous DNA for base pairs. And after that distance is covered and the double-stranded break replication has gone past the point where the double-stranded break was, the whole crossover structure is again resolved, and you result in 2 DNA segments, the original. Actually, it also involves some switching of DNA, but you end up with 2 strands of DNA that don't have any breaks present in them. And again, I might encourage you to refer back to these pictures or even try to find other drawings that represent how homologous recombination occurs, because it's a bit of a complex structures to really understand. The recombination genes are always expressed in the cell, indicating that breaks can occur at any time, and so they need to be there to fix it as quick as possible. Rec-A is a protein that is constitutively expressed, and it facilitates the process of recombination. What it does, and this is supposed to be a picture of Rec-A, kind of this bubbly-looking structure, is Rec-A bound to the DNA in red. And it binds to the double-stranded DNA and basically stretches it out. And as it does this, it kind of loosens up the DNA so that the single-stranded DNA from the broken strands of DNA can invade and utilize that DNA for base pairing. So Rec-A is very important for facilitating the recombination with homologous strands of DNA. If you lose the entire process of recombination, it leads to this disease called ataxia-telangiectasia. It's abbreviated AT. Now, AT is characterized by some physical characteristics that are easily identified. Telangiectasias, I'm not sure I'm pronouncing that properly, but those are basically like spider veins that are little red veins that show up often on the cheeks and on the ears, and sometimes in the white of the eyes. And AT also leads to problems with walking. As a person is developing, as a child, the walk will be very unsteady and not easily accomplished. And by the time the individual reaches the teens, they will be unable to walk altogether and have to utilize a wheelchair. And by the time the individual reaches the 20s, lifespan is not expected to last much longer. So recombination is really very important for the process of development of an individual. Whoops. And then, as I mentioned, there's also this process of repairing double-strand breaks called end joining. And this repairs double-stranded DNA gaps, basically by taking 2 DNA strands with ends and sticks them together and ligates them. And so it's good if this process happens to grab the DNA that was originally broken and stick those pieces back together. But it's also possible that this whole process might grab one broken end and then another chromosome altogether and add it onto the wrong chromosome. So it's potentially mutagenic, causing translocations and many other problems with breaking of DNA and trying to religate with an end joint, because there's no template. There's no way to really tell that it's doing this correctly. And it turns out that Deinococcus, which we talked about at the very beginning of the chapter, remember they have extremely fast repair mechanisms. They're also really good at end joining. So if they're exposed to radiation, very quickly they're able to religate their chromosomes so that if they're broken, they're able to facilitate religating and be able to survive real quick. Or not real quick, but real easily in response to the radiation. Okay, so let's just take a little break and look at what we know so far and what we have yet to cover before this class is over. So we've looked at types of DNA damage, and we've talked about adaoxoguanine several times, as well as thymine dimers. We've looked at repair mechanisms that are designed to fix the damage. Base excision repair fixes adaoxo-Gs. Nucleotide excision repair repairs thymine dimers. And we have learned that mutations in specific genes lead to diseases such as ataxia-telangiectasia. Nucleotide excision repair genes can lead to XP, and so on. Now this chapter is focusing on cancer. So what are specific genes that are involved with cancer, and how does DNA damage affect these specific genes and lead to the development of cancer? Well, to answer this, we're going to look some at the progression of the cell cycle and proteins that are involved with controlling the cell cycle progression. And I'm also going to look at some signaling pathways which serve to control the quality of the state of the cell in order to serve as checkpoints for allowing the cell to continue in its progression. Now, I'm sure in high school biology you learn about the process of the cell. So we're probably all familiar with interphase and mitosis of the cell. Well, just to cover it so we're all on the same base here, during interphase there's the growth phases indicated by G1 and G2. And then this DNA synthesis occurs during the S phase. And then division of the cell occurs during mitosis or M phase. Now, it turns out that there are proteins that were discovered to increase with concentration and decrease with concentration dependent upon what cycle the cell is in. And were called cyclins because they appear to cycle along with the cell. And so these lines here indicate, say, cyclin D starts to increase in concentration towards the end of G1. And then as the cell passes into the S phase, it decreases and isn't present again until a cell comes back to G1, and so on and so forth. So cyclin E also increases through G1, and so on. There are pathways that these cyclins control. Often they control kinases, which affect signaling pathways. So it's all very interacting, helping to promote the cell into the next pathway. So cyclin D and cyclin E help to promote the cell from G1 to S phase. Whoops, how did that happen? Let's just end this. Skip right to the end of the lecture. Okay. Hmm. 40? Okay. It doesn't seem to be responding at all right now, so. No, obviously not. So, oh, well, we're waiting. Any questions on anything before we move on? I appreciate all your comments from Friday. I read all through your note cards and things you had to say. I really appreciated it and found that it was very useful for just seeing things that I'm doing well and need to improve on. So thank you very much for your feedback that you were able to give. Well, now let's see if I did absolutely nothing. So we're looking for 40. Here we go. Okay, there we go. Now let's see if we can get this up in a slideshow format. Oh, okay, there we go. Well, this is just a picture showing some of the yeast cyclins that are involved with progression of the cell. And I just noticed a typo here. They're not lost, there's just lots of them. So, yes, we do know what they are, and we know where they are. They're inside the yeast. So you can see these numbers indicate some of the cyclins. Cyclins are often abbreviated CDC. The kinases that are affected by them are CDCs. You can see that there are just a lot of them, numbering all the way up to 48 or even higher, that are involved with progression of the cell. Each has a different role, and some of them may overlap in promoting progression from G1 to S to G2 to metaphase. And so obviously there are lots of them, and often it's very difficult to try to learn and remember all the numbers, especially because numbers and names don't always overlap between different organisms as well. But it's really quite amazing how they do play a role in progression of the cell. It turns out there are checkpoints along the way that basically serve as quality control. They're the guys that are standing there saying, now this is just a little example of how, say you start right here at the G1 checkpoint and it's asking, well, is the cell ready to go on? Is it big enough for replication to occur? Is it a favorable environment? Are we ready to start replicating DNA? And if that checkpoint is passed, then the cell is able to go on to S and replicate through the DNA, and so on and so forth. So each one, basically there are proteins that serve to regulate and observe what's going on and say it is time for the next phase or whatever needs to come next. And these work by observing growth signals. They look to see if there's damage in the DNA, because if the damage is present, we don't really want replication to go. Or if there's a lot of damage present, we don't want it to go because that could be potentially harmful to the cell. Also observing how long the telomeres are. If they're getting too short, then maybe replication isn't a very good process. Is the spindle forming properly? Can mitosis occur? And so on and so forth. If things are wrong or it's not the correct environment for things to occur, it'll stop the cycle. Or there's another spelling mistake, apoptosis may be initiated if it's really bad and things are totally wrong. Now just to cover a little bit on what apoptosis is, it's the process of the cell basically packaging up all its stuff and preparing to die. What it does is take all the contents, package them up into little membranes, and send them out so that they can be digested by macrophages. And it's this way of disposing of all the contents within the cell so that the cell can properly die. External factors can trigger apoptosis. Also internal factors such as this extreme DNA damage or checkpoints along the pathway that say things are just totally wrong. It's better just to quit altogether. Enzymes called caspases are involved with the process of apoptosis. They actually have this amplified cascade where one caspase activates another and they activate themselves and they serve to really amplify and activate a lot of enzymes all at once. So it ensures that the cell is for sure going to die because there's a lot of these enzymes that suddenly become active all at once. So apoptosis involves, basically this is called blooding, where you can see these little balls of membranes that are coming away from the cell. And there's contents from the cell inside there, ready to be digested by the macrophage. Now the checkpoints within the cell cycle are helped to regulate signaling pathways, and they activate them. And these pathways do involve kinases, which add phosphate groups onto proteins. They also involve phosphatases, which remove phosphates from proteins. And there are many other protein components which serve to propagate the signal on from one protein to another. So what happens is some signal might activate a pathway that leads to a response. And there are many different pathways. There's so many that often they get very confused, and it's actually pretty hard to understand which ones are involved in each pathway because some of these have extensive networks where they interact with many, like one protein might be a component of 4 different pathways. There's a lot of redundancy in functions for each of these cell signaling pathways. So it's difficult to assign specific activity for one pathway, but it's really pretty complex and pretty interesting as well. Basically, though, we can find that defects in specific pathways are linked to the formation of cancer. Now, an example is the DNA damage checkpoint, or the proteins that recognize damaged DNA. One of these is called ATM, also involved in the development of the AT, which is important, which, if you remember, we talked about during recombination. So ATM is a signal is a protein kinase that initiates a signaling cascade in response to the damaged DNA. So if there are proteins that serve to recognize damaged DNA, say this little orange star on the DNA, a sensor protein might come along and recognize that this is damaged. And then it activates, say, ATM, or another signaling protein or kinase, which activates other transducers, and then those serve to amplify the signal to activate many other things, such as the DNA repair enzymes for nucleotide excision repair or base excision repair. Transcription, maybe transcription should be halted for a while this is dealt with. Also, apoptosis proteins may be activated. So there's a lot of things that go along, but ATM is a big player in DNA damage checkpoint and says when it's time for the cell to activate repair of DNA. And it serves to activate two specific proteins that we're going to look at today called BRCA1 and P53. Now, BRCA1 and BRCA2 are two genes involved with breast cancer development, so that's why we have this abbreviation BRCA. Mutations in one of these genes, 1 or 2, will lead to a 70% chance for a woman to develop breast cancer. And ATM, since it's a protein kinase, actually phosphorylates this protein BRCA1. And it appears that this protein actually serves as a scaffold for the DNA repair and recombination proteins. So basically it's like a ladder standing there, letting the other proteins come in and bind to it so that they can be in a close context and able to facilitate repair or recombination of damaged DNA. If you mutate this gene, or the protein is non-functional altogether, the cell cycle control in response to DNA damage and repair of DNA is not able to function. And so that's how cancer may develop, because dysfunctional DNA repair, as you know, leaves the cell up to finding other ways to deal with it, leading potentially to cancer. And also there's evidence that BRCA1 binds to double-stranded DNA breaks and helps to prevent incorrect end joining between a double-stranded break and the incorrect DNA that it's trying to be ligated to. And there's still a lot to be known how that actually is facilitated through that protein. Another example of loss of cell cycle control is with the retinoblastoma protein. RB, as it's abbreviated, serves as a regulator to keep the cell in the G1 phase. As long as it's present at a high enough concentration, the cell cannot progress from G1 to S. And it does this by binding the transcription factor called E2F. And when it's bound to E2F, transcription cannot be occurred, so the S phase cannot initiate synthesis of other proteins that are necessary for the S phase. Retinoblastoma, eventually as the cell progresses from G1 to S, the concentration of RB decreases, and as a result, E2F activity is able to increase and promote S phase in the cell. RB is mutated in many cancers. It plays an important role with the development of many cancers, and is first discovered through retinoblastoma, which is also involved with the eye and development of cancer in the eye. Many children are born with a mutation in the RB gene already, and if they acquire another mutation that hits the other allele of the RB gene, it leads to eye cancer for the individual. There's also a later onset form of retinoblastoma, which would require that individuals born with both alleles fully intact require hits or mutations in both alleles before this cancer can develop. And as I said, we're going to also talk about this protein P53, and this plays a big role in a lot of cancer cells, and it's found to be mutated in at least half of all tumors. And again, the level of P53 affects what phase of the cell cycle the cell is in. So dependent upon the concentration, it also controls the cell cycle pathway. ATM, again, is a kinase that activates or that phosphorylates P53, and this leads to help delay the degradation of P53. And as long as P53 is present and not degrading, then it blocks the cell cycle from progressing to the next stage. This also buys time for DNA repair and for other enzymes that are needed to accomplish, say, recombination. Now, it turns out that P53 acts on a lot of different proteins, and they all play a role in perhaps if there's high levels of DNA damage. P53 activates this Bax protein, which serves to signal to initiate apoptosis of the cell. If there's only moderate levels of DNA damage, P53 activates another protein called P21, which eventually signals for the cell cycle to simply be arrested until the DNA damage is repaired. And there's a lot of other proteins that help to regulate the level of P53, such as MDM2. So it's very important for the development of cancer and the prevention of cancer, and also known to play a big role in the development of tumors. And I'm going to just skip this slide because it shows basically the same thing, that P53 is integral to the development of perhaps cancer or apoptosis, especially if P53 is inactivated. Now, it turns out that P53 and RB are both extremely important for cell development. In fact, you need both P53 and RB pathways to be lost to allow tumor development. So there are proteins that serve to censor like inappropriate cell activation or DNA damage. If RB is inactivated, then there's still P53 to compensate for the activity of retinoblastoma protein, because they both function shutting down the cell cycle during DNA damage. And there are other protective pathways, but if you knock out both P53 and the retinoblastoma protein, tumor development is very likely to occur. And just quickly, we're going to hit on another topic called tumor angiogenesis. And we talked about this as one of the hallmarks and one of the signs of cancer. That's easy to see. Now, as I mentioned, tumor cells can only grow a certain size before they need additional blood supplies in order to provide enough nutrients for the cell to continue to grow and develop. And so it recruits the development of new blood vessels to come close and to thus bring more blood and more nutrients to the tumor so that the tumor can grow even larger. And so this is the process of angiogenesis, which is the growth of new blood vessels. And metastasis is the process by which cancer cells can pick up and move or spread from one area in the body to another. And as angiogenesis occurs and more blood is provided to the cells, it also provides more opportunity for cancer cells to pick up and move through the bloodstreams to other locations in the body, and that is metastasis of the cancer cells. So, just in review, some characteristics of cancer: genome instability. Mutations in the genome, very, very likely and very, very common in cancer cells. Self-sufficiency of growth signals. They might produce their own signals saying, keep growing and keep dividing so that we keep growing and form this tumor. Also, cancer cells are insensitive to growth inhibition. Often they don't die when they should, so they don't go through apoptosis at the time that they should. And they might continue replicating at a limitless, without any limit. So senescence is the process by which a cell basically kind of stops growing and kind of doesn't do anything more. And they do not reach senescence when it's a tumor or a cancer cell. Angiogenesis and tissue invasion and metastasis. So though these are very vague, big concepts, there are many, many factors that play a role in each one of these. Basically, if you can see that the cell has these characteristics, you can look at it and say, yep, it's cancer. Just like this early microscope might help you recognize what a mammoth is. I think I'm going to go ahead and stop right there. So I thank you very much for your attention. This is my last lecture, so I appreciate your respect and time for attending.